Hello everyone, in this video we will talk about siRNA, shRNA and miRNA. That means siRNA means small interfering RNA, shRNA means short hairpin RNA, and miRNA means microRNA. These RNAs I'm talking about, these are all non-coding RNAs, okay? Non-coding RNAs with important role in gene regulation. They have recently been investigated as novel classes of therapeutic agents for the treatment of wide range of disorders, including cancers and infections. I have already discussed about RNAi mechanism in my previous video, and you can check my previous video if you want to know more about the RNAi mechanism or you want to understand the basic concept of RNAi mechanism. Actually, RNAi mechanism is a natural process that silences gene expression by promoting the degradation of mRNA. Whenever there is a double-stranded molecule in the cell cytoplasm, then your RNAi mechanism come into play in order to suppress, some expression of some genes, particular genes. Okay, RNAi mechanism for gene silencing. Whenever there is a double-stranded RNA in your cytoplasm. So let's see what is the miRNA and how it acts on the cell for, in order to induce gene silencing mechanism. Your miRNA is produced whenever a gene is transcribed by polymerase II enzyme to produce an mRNA molecule. Okay, if this mRNA molecule have some sequence complementarity, if it's very long and it has repeated sequences, or it has some base sequence complementarity, then they will form some structures like this. Okay, the sequences which are complementary to each other, they will fold and bind to each other, forming this type of stem loop structure. Okay, this structure is formed in the nucleus, inside the nucleus. Okay, this is nucleus. Inside the nucleus, this type of structures are formed, and this is known as your primary mRNA, miRNA, or pri-mRNA, miRNA. This structure is then processed by two enzymes known as DROSHA and PASHA. They are used as a microprocessor here. So your PASHA is also known as DGCR8 molecule. Okay, and this, what they do, this DGCR8, which is known as Di-George syndrome critical region 8, it orients the catalytic RNase domain of DROSHA molecule to liberate hairpins from pri-mRNA molecule, and thus it forms a structure like this. And this is known as your precursor miRNA molecule. Okay, this all things happened inside the nucleus. Now this molecule is transported to the cytoplasm, okay, with the help of the protein called exportin 5. This will transport this precursor miRNA molecule inside the cytoplasm. Now this molecule is again processed by the enzyme dicer present in the cytoplasm, which is a RNase 3-like enzyme that will act on this strands, and it will separate this loop from this preliminary miRNA molecule and form a structure like this. This will have a 3-2 nucleotide overhang on both the strands. Okay, this structure is known as miRNA duplex. Now this duplex molecule will interact with the RISC protein enzyme, which is a multiplex protein. Okay, this RISC protein consists of various types of it's a multi protein enzyme which have slicer activity. It will have slicer activity, and there is a protein family called argonaut protein family that will help in recognizing the 3' end of the this mRNA duplex, and it will bind to the RISC protein. Okay, suppose this is your RISC protein, then your mRNA duplex will bind here. Okay, this RISC protein has a helicase activity that will help in unwinding of this double-stranded mRNA duplex, and it will release the passenger strand or the sense strand that is not having complementary sequence with the target strand. You can say it's a passenger strand. Okay, passenger strand or sense strand. Okay, that will be released. Now the RISC protein is only having the single stranded miRNA molecule that is having sequence complementary with the target mRNA molecule, whose expression is going to be stopped. Okay, so this is the antisense strand because it will have sequence complementary with the sense mRNA molecule or target mRNA molecule. Now this miRNA molecule will guide, this is known as guide strand, because this will guide this RISC protein to bind to the target mRNA molecule. Suppose this is your target mRNA molecule and this will bind here. mRNA molecule will not have complete sequence similarity with the target mRNA molecule. Okay, it will have some mismatch sequence complementarity. Okay, so this will ultimately result in degradation of the mRNA molecule or cleavage of the mRNA molecule, or it might also repress a translational process, because of which no protein will be formed from this mRNA molecule. No protein is formed. As a result, it will cause gene silencing. So we can see here that a double-stranded RNA molecule is formed here, forming a stem loop-like structure, which is processed by Drosha and Pasha enzyme to form a precursor mRNA molecule, which is transported to the cytoplasm here, and then this is processed by the enzyme, which is an RNase tree-like enzyme that will cleave the stem loop structures and form a structure that will have a 2 nucleotide 3' overhang, and this is known as mRNA duplex. Then what will happen? This will interact with the RISC protein, which is a multi-protein complex, and it has various activities, like it has helicase activity, it is having slicer that will that is having RNase activity to cleave the target mRNA molecule. Then there is a protein family called argonaut protein family that help in recognizing the 3' protruding end of the mRNA complex, and then this will then mRNA molecule, the guide strand of the mRNA molecule will direct this RISC protein to bind to the target mRNA molecule, and then ultimately RNAi activity of the RISC protein will cleave this mRNA molecule. Okay, so this is about your miRNA mediated gene silencing. Now we'll study about siRNA. Your siRNA may be produced inside the cell from cellular genes, or it may be produced from infected pathogens, or sometimes it can be introduced artificially inside the cytoplasm. Okay, then after production, suppose if it is produced by RNA polymerase II to form mRNA, okay, this mRNA, then it will be transported to the cytoplasm, where it will form a double-stranded structure. It's a long double-stranded structure, maybe 100 to 120 nucleotide long, then this will be processed by dicer molecule to form small structures like this. This will also have 2 nucleotide 3' overhangs. Okay, now the process is similar. It will interact with the RISC protein molecule, okay, to come and join with the RISC protein molecule here. Suppose this one is a RISC protein, okay, this will bind, these 2 strands will bind. But the difference with the miRNA is that in case of miRNA, the helicase activity will cause unwinding of this double helix and will release the passenger strand. But here it is not that case. Here what happens, the Argonaute family of RISC protein is having cleavage activity which will cleave the passenger strand. This will degrade the passenger strand and leaving behind only the guide strand in the RISC protein. Okay, only the guide strand will be there because your passenger strand will be completely degraded, unlike your miRNA where the passenger strand is released, not degraded. Okay, with the help of your RNase activity, this passenger strand is degraded here. Now similar case, your RISC protein will be directed towards the target mRNA molecule. Okay, this is your target mRNA molecule. Now this passenger strand of the miRNA will direct this RISC protein to bind to this target mRNA molecule, and this will cause cleavage of the mRNA strand. Okay, so your expression of that particular gene is blocked, and now triggering gene silencing. So we can see that siRNA and miRNA mediated gene silencing mechanisms is very much similar. The only difference is that they have structural dissimilarities or differences, and the major difference is that siRNA molecule has incomplete sequence complementarity. Okay, because of this region they can have multiple target mRNA molecule, but your siRNA molecule have complete sequence similarity with the target mRNA molecule. Therefore they are highly specific, which target only one mRNA molecule. Okay, single mRNA molecule. Now comes your short hairpin structure or shRNA molecule. Okay, actually what happens, siRNA molecule may be introduced directly into the cell cytoplasm, okay, where they can undergo the normal gene silencing mechanism of the cell, but they can be alternatively introduced into a vector which form a short hairpin like RNA, and then it can be introduced into the cell nucleus, like if it's a vector kind of vector that is coding for shRNA. Okay, that is coding for shRNA, a vector molecule, typically through delivery of a viral vector. Suppose this is a viral vector molecule that will be introduced into the cell's nucleus. Suppose this is your nucleus. Then this DNA, your vector, will be transcribed to form 2 types of mRNA molecule. Okay, 2 types of mRNA. One will be your microRNA adapted shRNA, and the second one will be your simple hairpin shRNA. The structure will be like this. This one will have structure like microRNA, and this one will have a small hairpin like structure. Okay, so since this structure is like micro miRNA, okay, this has to be processed in order to be transported through the nuclear pore. Okay, this is similarly processed by Drosha and Pasha to form a small hairpin-like structure, and now both of these are exported by exportin 5 to the cytoplasm. Now its processing mechanism is similar with siRNA and mRNA, because this will be cleaved by dicer molecule and then will be loaded in the RISC protein, okay, and then the guide mRNA will direct this RISC protein to bind to the target mRNA molecule, and that will result in cleavage of the target mRNA molecule, resulting in gene silencing. Okay, the rest of the phenomenon is similar with siRNA and mRNA, but the thing is that it will be processed inside the nucleus to form two different types of shRNA structure. One will be similar to miRNA and the other will be short hairpin like structure. The one which is similar to miRNA will be processed and exported to the cytoplasm, okay, with the help of Drosha and Pasha, and this type of structure is transcribed by RNA polymerase 3, and this type of structure is transcribed by RNA polymerase 2. Okay, so this is how siRNA, miRNA and shRNA are involved in gene silencing mechanism. Thank you for watching this video.